Welcome back to the deep dive. Today, we are actually dismantling a metaphor that I I think it holds a lot of us back.
Oh, for sure.
We tend to think of the vascular system, our arteries and veins, as just plumbing, like
Yeah.
passive pipes,
Yeah.
just these PVC tubes moving red liquid from point A to point B.
Right, which is, that's about as accurate as calling a supercomputer a calculator. It
Yeah.
just completely misses the entire point of what the system actually does.
Exactly, because I've been going through the medical texts and those histological slides you shared, the stack labeled vascular.pdf.
Yeah, the deep anatomy stuff.
Yeah, and the complexity here is just staggering. We aren't looking at static tubes at all. We're looking at a dynamic, responsive organ system that spans the entire body. It thinks, it reacts, and as we'll see later, it even rebuilds itself.
It really is an engineering marvel. If your household plumbing could grow new pipes when you added a room, or actively decide to shut off water to the kitchen to, boost pressure in the shower because it knew you were dirty, then maybe we could call it plumbing. But the vascular system is doing that constantly. It's a living pipeline.
So our mission today for you listening is to basically decode that system.
Yeah.
We're going to move past that grade school, red equals oxygen, blue equals deoxygenated mindset.
Oh, please, yes. Let's
And-
kill that myth.
We want to understand the actual architecture of these vessel walls, the physics of these startling forces that keep us from swelling up like balloons, and the incredible way the body stores and shifts your blood volume around.
And it all really starts with the map. The fundamental rule of the cardiovascular system is that it's a closed loop.
Right.
It begins and ends at the heart.
Yeah, and I think most of us know the basic beat, right? You got
Yeah.
the right atrium, drops to the right ventricle, out to the lungs, back to the left atrium, left ventricle, and boom, out to the body.
Yep, that's the grand loop.
But looking at the source material, that color coding rule is where people just get tripped up immediately.
It's the classic trap. We're taught from day one that arteries are red and veins are blue.
Always,
But the physiological definition actually has absolutely nothing to do with oxygen content.
right?
It's strictly about direction relative to the heart. So arteries carry blood away from the heart, veins carry blood toward the heart.
Which explains the whole pulmonary circuit exception.
Precisely. Let's trace a single blood cell for a second. It enters the right atrium, drops through the tricuspid valve into the right ventricle. Now, the heart squeezes. That cell is blasted through the pulmonary semilunar valve into the pulmonary arteries.
And at this exact point, the blood is oxygen poor. It's technically, quote unquote, blue.
Correct. But because it is leaving the heart, it is in an artery.
Got it.
It goes to the lungs, picks up oxygen, turns that bright crimson red, and then returns to the left atrium via the pulmonary veins.
So right there, you have veins carrying highly oxygenated blood. It completely flips the script.
It really does.
So once we clear that hurdle and the blood travels through the bicuspid valve into the left ventricle and is blasted out into the aorta, we enter the systemic circuit.
Right. Out to the rest of the body.
And this is where the architecture gets really, really interesting to me. I always assumed a blood vessel was just, I don't know, a single layer of material, like a rubber hose.
Most people do.
But the histology creates this much more complex picture. We're actually talking about 3 distinct layers, or tunics.
Correct. Whether it's a massive artery like the aorta or just a medium-sized vein in your arm, they share this exact same 3-layer blueprint.
Okay.
It's the proportions that change based on what the vessel needs to do. So think of it like a sandwich. The innermost layer, the one actually physically touching the blood, is the tunica intima.
Which the text describe as being incredibly smooth.
Oh, it has to be. It's lined with endothelium, which is just a single layer of squamous cells.
Like flat scales.
Exactly. You want this surface to be like Teflon, completely frictionless. Because if it's rough, blood cells start bumping and sticking, platelets get activated, and bam, you get clots.
Which is bad news.
Very bad news. And in arteries, right underneath that Teflon layer, you also have this internal elastic lamina.
I was actually looking at the slides for that. It looks like this wavy, undulating line under the microscope.
Yeah, that waviness is highly functional. It basically allows the vessel to stretch and recoil without tearing apart.
Fascinating.
Then you move outward to the real meat of the sandwich, the tunica media.
The middle layer.
Right. This is the muscle layer. It's made of circular, smooth muscle and sheaths of elastin fibers.
And this is where the actual control happens, right? Like, this isn't just structural scaffolding. This is functional.
Oh, absolutely. This is the command center for your blood pressure. When your sympathetic nervous system fires, this smooth muscle contracts.
That's vasoconstriction.
Exactly. The lumen in the opening gets smaller, so pressure goes up. When it relaxes, you get vasodilation. And the actual thickness of this muscle layer dictates how much control your body has over that specific vessel.
Okay, and then finally we have the outer wrapper, the tunica externa, or they also called it the adventitia.
Yeah, mostly made of collagen fibers. It's the anchor.
So it keeps things in place.
Right. It keeps the vessel tethered to the surrounding tissue so it doesn't just flop around inside your chest cavity or your leg when the blood pulses through. It protects and reinforces the whole structure.
Okay, stay in the structure for just a second. I found a detail in your notes that I honestly had never even considered before. It's this concept of the vasa vasorum.
Ah, yes. Latin for vessels of the vessels.
It sounds incredibly meta,
It does.
but when you actually think about the physics of it, it makes total sense. Because the aorta has these thick walls to withstand all that pressure from the heart.
Right. And here is the fundamental problem. Cells need oxygen to survive. The wall of the aorta is so incredibly thick that the oxygen and nutrients flowing inside the main tube cannot diffuse deep enough to reach the outer layers of the vessel wall itself.
So the cells on the outside of the pipe would literally starve.
Exactly.
So the pipe is so big
Yep.
that the outside of the pipe needs its own tiny plumbing.
That's it, exactly. The body actually grows a network of tiny capillaries, the vasa vasorum, inside the tunica externa, just to feed the smooth muscle of the large artery.
That is wild.
It really highlights that these vessels are living tissue. They have metabolic needs just like your liver or your brain. They aren't inert rubber tubes. They are hungry tissue that needs to be fed constantly.
That is such a cool detail. So we have this 3-layer blueprint, but the source material makes a really big distinction between the different types of arteries.
It's a classic case of form following function.
Right. And we start with the elastic arteries.
These are the heavy hitters. The aorta and its major branches immediately leaving the heart. Their primary job is simply to deal with the violence of the heartbeat.
Violence is such a good word for it. Because the heart isn't a continuous pump like a sink faucet. It's explosive.
It's boom, rest, boom, rest. If your arteries were rigid pipes, like lead pipes in an old house, your blood pressure would skyrocket to dangerous levels during the beat.
During systole.
Right, and then it would drop to near zero in between beats during diastole.
So you just pass out between every heartbeat because the flow would completely stop.
You would. It would be a nightmare.
That is a terrifying image.
It really is. So the elastic arteries are loaded with elastin in all three tunics. They act as pressure reservoirs. When the heart pumps, they expand like a balloon to absorb all that massive kinetic energy.
Right.
Then, while the heart is resting and refilling, they snap back. That elastic recoil literally pushes the blood forward while the heart is pausing.
So they basically turn a jagged, explosive rhythm into a nice, smooth stream.
Exactly. By the time the blood actually reaches your organs, it's a steady flow, not a jackhammer.
But as we move further away from the heart, the strategy changes. We transition to muscular arteries.
Right, because now we need to direct traffic.
So here we trade elasticity for control.
Correct. These vessels have a massive tunica media. We're talking up to 6 layers of smooth muscle relative to their overall size.
Wow.
Their job is distribution. They are the traffic cops deciding which organs get blood at any given moment. If you need more blood in your stomach to digest a big meal, these are the arteries that dilate to make that happen.
And then the road narrows down even further to the arterioles. The notes actually call these the gatekeepers.
That is the perfect term for them. Arterioles are the smallest arteries. Sometimes you'll hear them called precapillary arterioles.
Okay.
They control the flow directly into the capillary beds, and they are the primary creators of resistance in the entire system.
So if the arterioles clamp down, the shop is closed.
Essentially, yes. If the arterioles leading to a specific patch of tissue constrict, no blood gets into that tissue.
Which makes sense.
This is critical for things like temperature regulation. When you're cold, the arterioles in your skin constrict to keep the warm blood deep inside your core.
Okay, so we've navigated the highways and the side streets, and now we arrive at the final destination, the capillaries.
The main event.
To me, this is where the real action is. The anatomy here is fascinating because it's so reductive. They just strip everything away.
They have to. The capillary's only job is exchange. You cannot trade oxygen for carbon dioxide through a thick, heavy muscle wall.
Right.
So the body strips the vessel down to just the tunica intima, just a single layer of endothelium and a sparse little basement membrane.
And the scale is tiny. The notes say red blood cells literally have to march through single file.
They actually have to fold slightly to fit. It's that tight.
Wow.
But this maximizes the surface area of the cell touching the vessel wall to make that gas exchange as fast and efficient as possible.
But even though they are really simple, they aren't unguarded. The text highlights these little cells wrapped around the capillaries called pericytes, and they sound like they've been overlooked for a really long time.
Oh, they definitely have been. For years, histologists just saw them as structural, like little tiny clamps holding the capillary together. They kind of look like spiders hugging the tube.
Spiders hugging a tube, great visual.
But we now know they are massive, multitasking powerhouses.
The source mentions they look like embryonic mesenchymal cells, which sounds highly significant.
It is deeply significant. It gives a massive clue to their actual function. They do support the structure, yes, but they also help regulate blood flow right at that microscopic level.
Right.
And crucially, in the brain, they are the key architects of the blood-brain barrier, keeping toxins out of your delicate neural tissue.
But the most mind-blowing part for me was their regenerative potential. The notes say they can differentiate.
Yes. Parasites are stem cell-like. If there is an injury to the vessel or the surrounding tissue, they can detach, migrate, and transform.
Transform into what?
Well, they can turn into fibroblasts to create scar tissue or even smooth muscle cells to repair larger damaged vessels.
So they're essentially a dormant microscopic repair crew sitting on the walls of your capillaries, just waiting for a distress signal.
That's a fantastic way to put it. It completely recontextualizes the whole system. It comes with its own maintenance staff built right into the walls.
Now, looking at the capillaries themselves, I noticed the notes say there isn't just one type. It really depends on where you are in the body.
Right. It entirely depends on how much leakiness you need in that specific organ.
Okay, so first you have continuous capillaries.
These are the strictest ones. The endothelial cells are joined by tight junctions. You find these in your skin and your muscles.
And in the brain, right.
Exactly. In the brain, these junctions are incredibly tight, forming that blood-brain barrier we just mentioned. They let water and small solutes pass, but they keep the big stuff like cells, bacteria, and large proteins safely inside the bloodstream.
Then you have the fenestrated ones.
Right. Fenestra is Latin for window.
So they have actual windows.
Basically, yes. These capillaries are riddled with pores. Imagine a garden hose that someone took a tiny hole punch to. You find these anywhere you need rapid exchange or filtration.
Like the kidneys.
Exactly. The kidneys are constantly filtering your blood to make urine, so they need very high flow. Or the small intestine, where you're absorbing massive amounts of nutrients from your food. You need those windows to facilitate that rapid movement.
And finally, you have the sinusoids. These sound like the absolute Wild West of the vascular system.
They really are. Sinusoids are highly modified, incredibly leaky capillaries with large, very irregular lumens.
And where are these?
They are found in the liver and the bone marrow. They have massive, gaping gaps between the cells.
But why would you want a vessel that leaky? Wouldn't you just bleed out internally?
Think about the bone marrow for a second. That's the factory where your new blood cells are physically manufactured.
Yeah.
If you had a tight, continuous capillary there, it would be a prison. The brand new red blood cell couldn't actually get out into the circulation. You need a sinusoid with a huge gap so that cell can literally slip through the wall and enter the bloodstream.
Oh, that makes perfect sense. So we have the structure, we have the types. Now we really have to talk about the physics.
The fun stuff.
The source material brings up Starling forces. And this is where I think a lot of people might get lost. But it's essentially just a physical tug of war, right?
That is exactly what it is. It's a battle between two competing pressures that determines whether fluid leaves the capillary or gets pulled back in.
So on one side, pushing fluid out is filtration.
Right. This is driven by capillary hydrostatic pressure, which in plain English is just blood pressure.
Right, the mechanical force.
Exactly. The heart is pumping, pushing fluid physically against those leaky capillary walls. That mechanical force pushes water and small dissolved nutrients out into the surrounding tissue to feed your cells.
Okay, but if that was the only force acting on the blood, all the water would just leave our vessels. Our tissues would swell up like water balloons, and our blood pressure would completely crash to zero. We'd dry out from the inside.
Yes, it would be catastrophic. So nature provides a brilliant counterforce. Reabsorption.
And this is driven by osmotic pressure.
Yes. And this is where the plasma proteins come in to save the day.
Albumin, specifically, according to the notes.
Yep. Albumin. These are large proteins that are permanently stuck inside the capillary because they are just too physically big to leak out of the pores.
Okay.
Now remember basic high school chemistry. Water loves to go where the solute concentration is high. It acts like a magnet.
Right.
The proteins trapped inside the vessel act as a magnet and constantly pull the water back in.
So it's a delicate balance. At the start of the capillary bed, the blood pressure is high from the heart, so it overpowers the proteins and pushes the fluid out.
Correct. And by the time the blood reaches the end of the capillary, that physical blood pressure has dropped significantly because of friction.
So the protein magnet suddenly wins.
Exactly. The osmotic pull wins, and it pulls the now waste-filled fluid back into the vessel to be carried away.
And what happens if that balance breaks down?
You get edema, severe swelling.
Oh.
This is actually why people with severe starvation get those tragically distended bellies, a condition called kwashiorkor.
Because of protein deficiency.
Exactly. They simply don't have enough protein in their blood to generate that osmotic pull, so the fluid leaks out into their abdomen and just never comes back in.
Man, that is a stark reminder of how incredibly precise this system has to be. It's a delicate mathematical equation happening perfectly in billions of vessels every single second.
Absolutely. And when it works perfectly, you don't even notice it's happening.
Now, earlier we established that this isn't just plumbing because household plumbing doesn't grow,
Right.
but this system does. The text distinguishes between two specific growth processes, vasculogenesis and angiogenesis. Now, I feel like these terms get used interchangeably in the news a lot, but they are actually very distinct.
They're fundamentally different mechanisms. Vasculogenesis is the de novo formation.
Meaning from scratch.
Yes. It's creating something from absolutely nothing. This mostly happens when you're a tiny embryo. You have these precursor cells called hemangioblasts that migrate to where they are needed and literally form the very first vascular web.
So that's laying the initial foundation.
Yeah.
And then what about angiogenesis?
Angiogenesis is what happens in the mature organism. In you and me sitting here right now, it's the sprouting of new vessels from existing ones.
Like growing a new branch off an already existing tree.
Exactly like that.
And this is driven entirely by need.
Almost entirely, yes. If you pack up and move to a high altitude where oxygen is thin, your body senses that hypoxia.
The lack of oxygen.
Right. It triggers the release of a chemical signal called VEGF, vascular endothelial growth factor. That signal basically tells the existing vessels, hey, we're starving here, build more roads.
And the mechanism the text describes is super complex. It involves degrading that basement membrane we talked about earlier, migrating cells, and physically forming new tubes.
It's an elaborate cascade. You also need something called angiopoietin-1 binding to Tie2 receptors just to stabilize the new vessel once it forms. It's a biochemical choreography that is incredibly strictly regulated by the body.
But there's a dark side to this, too. The notes mention cancer.
Yes. And this is actually one of the most critical areas of modern cancer research.
Really?
Because a tumor is essentially just a ball of rogue, mutating tissue. But as it grows larger, the cells deep in the middle get too far away from the existing blood supply. They start to starve and suffocate.
So they basically hijack the system.
They absolutely do. They release massive, unnatural amounts of VEGF. They trick the body into performing angiogenesis. The tumor literally forces your body to grow a custom blood supply just to feed its own expansion.
Which is why, as I understand from the reading, many modern cancer drugs are classified as angiogenesis inhibitors.
Exactly. Drugs like Avastin, they work by actively blocking VEGF. The strategy is to starve the tumor to death by cutting off its supply lines before they can even form.
Wow.
It's the exact same biological tool your body uses to heal a scraped knee, just weaponized by the cancer.
That is profound complexity. The exact same mechanism that saves you can kill you, completely depending on the context.
Context is absolutely everything in human physiology. The body doesn't judge good or bad, it just responds mechanically to chemical signals.
Well, let's shift gears to the final piece of the puzzle, the fluid itself. Or rather, where the fluid is physically hiding at any given moment. I saw a chart in the source material that completely surprised me. It broke down total blood volume distribution at rest.
Let me guess, the venous volume shocked you.
Yes. I always assumed it was roughly 50-50, half in the arteries pushing out, half in the veins coming back.
That's what most people think.
But the chart says 60% of our total blood volume is just sitting in the systemic veins and venules at any given time.
That is exactly why we call veins capacitance vessels, or blood reservoirs. Remember we said their walls are thinner and much more compliant?
Right, less muscle.
They can physically stretch to hold a huge amount of extra volume without the pressure shooting up to dangerous levels.
The notes mention the abdominal organs and the skin specifically as major reservoirs. And the spleen, he says the spleen alone can hold nearly a liter of blood.
The spleen is like a highly specialized storage tank. In some animals, like dogs or horses, it's even more dramatic. They actually squeeze their spleen to dump extra red blood cells into circulation when they chase a rabbit or run fast.
Wow.
In humans, it's not quite that dramatic, but it's still a very significant reserve. This distribution is critical because it gives the human body a massive backup supply on demand.
Right, because that 60% figure is only true when we are just resting on the couch.
Correct. This introduces the exercise shift.
Yeah.
When you start heavy exercise, your cardiac output, which is the amount of blood the heart pumps per minute, changes drastically. At rest, it's about 5 liters per minute. But during maximum exertion, it can jump to 25 liters per minute.
That's a 5-fold increase in volume. That's the text called the cardiac reserve.
Exactly. But you can't just pump 5 times more blood everywhere equally. You don't have enough blood for that. You have to prioritize.
Right.
This is where the autonomic nervous system steps in. It triggers the sympathetic fight or flight response. It actively vasoconstricts the arteries leading to the digestive system and the kidneys.
It's like saying, sorry, stomach, you don't need to digest lunch right now. I'm literally running from a bear.
Precisely. It shunts blood away from those venous reservoirs, literally squeezing it out of the abdominal veins, and rapidly directs it toward the active skeletal muscles, the heart itself, and the skin to cool you down.
It's amazing to think about that level of rapid coordination. It's not just the heart beating faster, it's thousands of little arterioles opening and closing in perfect concert to completely reroute the entire traffic system of the body in a matter of seconds.
And that highlights exactly why vascular health is so critical and important. We tend to focus entirely on the heart.
Right, cardiovascular disease.
But the flexibility of this vascular tree, its ability to quickly dilate, constrict, and remodel itself, is just as vital. If your vessels become stiff or plaque-ridden over time, you lose that adaptability.
They lose the reserve.
You lose the reserve, exactly.
So to try and bring this all back together for everyone listening, we've gone from the thick elastic walls of the aorta that act as giant shock absorbers,
Down through the muscular arteries, actively directing traffic.
past the tiny arterial gatekeepers creating resistance.
And finally into the microscopic capillary beds, where those parasites watch over the massive exchange of life-sustaining fluids, driven entirely by that delicate physical battle between hydrostatic and osmotic pressure.
And all of this is constantly dynamic. It grows when we need it, shrinks when we don't, and serves as a massive fluid storage tank that can mobilize to save our lives in an instant. It really does completely change how I see the quote-unquote plumbing.
Oh, absolutely.
It's much more like a smart living city infrastructure than just a set of lead pipes.
It really is.
Mm.
And understanding this explains so much about our daily human experience.
How so?
Well, it explains why you get lightheaded if you stand up too fast. That's simply a lag in your venous reservoir adjusting against the force of gravity.
Oh, wow.
It explains why you can't digest food well while you're highly stressed. The blood has literally been shunted away from your gut. The deep anatomy dictates our everyday physiology.
And that brings me to the thought I want to leave our listeners with today. It goes all the way back to those parasites we talked about.
Ah, the microscopic repair crew.
Yes. We learned that these tiny, inconspicuous cells sitting right on our capillaries can literally turn into stem cells. They can become muscle. They can become scar tissue. If we are literally covered head to toe in microscopic repair crews just waiting for a chemical signal, what does that actually imply for the future of medicine?
That is the million-dollar question right there.
Right. Are we close to figuring out how to just, I don't know, switch them on to organically heal a heart attack or regenerate a damaged brain? The raw potential sitting inside our own vessel walls seems absolutely staggering.
And it is staggering. The ultimate cure for some of these things might not actually be a synthetic drug we introduce.
Right?
It might just be a specific signal we learn to send to the cells that are already waiting right there. We just need to learn how to speak their language.
Man, that is something to think about next time you feel your own pulse. Well, thanks for taking this deep dive into the vascular system with us today.
Always a pleasure. Stay curious.
We'll see you next time.